So guys, welcome to this lecture. Today we're going to be looking at the T-cell receptor and the B-cell receptor. So we're going to be looking a lot of gene rearrangement, generating the T-cell receptor diversity. So we discussed B-cell receptor diversity in a previous lecture, so if you guys want to check that out, we discuss how we form our B-cell receptor and our antibody. Today we'll be looking at a T-cell receptor, and here's just a brief overview of the structure if you want to take a look. So as we mentioned, we have our alpha, beta, and gamma delta. So we're going to start with our alpha, beta T-cell receptor. So there are two chains which make up each T-cell receptor, alpha, beta chains, or the gamma delta. These are linked by the disulfide bonds. You see here, two sulfides, and are part of the immunoglobulin receptor superfamily. Each chain has a variable and constant region, and there's an interaction between the variable regions of the alpha, beta chains, is where the antigen binding site occurs. Here's our alpha, beta chains, interaction of the variable regions. This is our binding site. And we also have a transmembrane domain, as we can see, and this is not secreted, unlike the B cell. So just keep this in mind. We'll discuss this later. Again, two classes of T-cell receptor are alpha, beta, gamma, delta. So you can see that they're pretty much similar in structure, other than they use different chains. And this is, you'll see, kind of like B cells, where there's the mu and delta, gamma, and epsilon to determine the isotype. These are similar in the sense that they're involved in the rearrangement. So each T cell expresses only one type of T cell receptor. So we either have alpha, beta, or gamma, delta, and not any combination of the both. And these are generated by gene rearrangement. So a single T cell produces a T cell receptor with only one antigenic specificity. As we discussed, same with B cells, they can only respond to the certain antigen. The T cell receptor is made up of two types of protein chains and uses immunoglobulin domain fold. That's why it gets that immunoglobulin superfamily designation. So now we're going to move into T cell receptor diversity. So T-cell receptor diversity in T cells is generated by essentially the same process as antibody in B cells, which we discussed in detail. So here we had our V, J, Z, and again, here's our gamma locus and alpha chain locus. And we got the same type of structure here where we'll see rearrangement will occur. They will have a randomly chosen variable region, a D region, and a J genes, which will combine to form an RNA transcript, which will then is translated into a protein chain. So here's quite complex figure looking at the diversity. So again, same process. You'll see that there's one alpha and one beta chain, or one gamma or one delta chain, which are assembled into the T-cell receptor. The T-cell receptor is not secreted. Again, B-cell receptor can be a membrane bound or secreted as antibody, but T-cell has to be membrane bound, and no class switching occurs. So once it gets this diversity, it stays that way. So organization of the T-cell receptor alpha chain. So here we can see our alpha chain locus. There are approximately 70 V alpha genes, 61J, and one constant. They have similar organization and gene arrangement process as immunoglobulin light chain genes. And again, these rearrangements generate diversity. Once a variable alpha gene segment joins with a J alpha gene segment, we have our V to J joining, which encodes the V region of the T-cell receptor alpha chain, which we have here. The T-cell receptor gene segments are arranged in a similar pattern to immunoglobulin. Gene segments are rearranged by the same enzymes. So we've talked about REG1 and REG2 and the VDGA recombinase. So those are still involved. So now we're looking at the alpha and delta chains. So a unique feature for T-cell alpha chain genes are interrupted by the T-cell gamma or delta chain genes. So the delta chain genes are located between the variable alpha and J alpha genes. There are approximately 6 functional delta gene segments, 3D, 3J, 4J, and 1 delta. So we can see here, here's our V, an alpha chain, as we can see here. So again, they're use the same enzymes. So here's just another look at the process. So the T-cell receptor commitment, alpha chain versus delta chain. So how is this decision made? If a variable alpha segment is joined by a J alpha segment, the intervening region is excised, as we can see here. This is where Excises. The excision of the delta chain gene segments means the T cell has committed to producing an alpha beta T cell receptor and therefore cannot produce gamma delta. Once it's chosen, is divided into this, it cannot go back or change. So beta chain. So 52 variable beta, 2D, 13J, 2C. The J and C segments, the D, J, and C segments are arranged in two clusters. Otherwise, it's a similar organization and gene rearrangement process as immunoglobulin heavy chain genes. So we have one D beta genes joins with a 1J gene, and one variable beta gene joins with the DJ beta genes, forming the VDJ joining, which encodes the variable region of the T cell receptor. Sorry, these letters get hard sometimes. But yeah, here's a figure. Again, we can see our number, approximately 50, 6, and 6 for J and beta. So again, here's pretty much a go-to figure if you want to just follow the recombination down. Here we can see our alpha rearrangement, beta rearrangement, and then our joining of the alpha beta, forming the T cell receptor. And again, here's our disulfide bond, transmembrane portion, and is not secreted. So gamma chain. There are 5 gamma, 5V gamma, 5J gamma, and 2C gamma. The JC genes arranged in two clusters, similar to the other one, otherwise similar in organization and rearrangement to the immunoglobulin light chain genes. One variable gamma gene joins with the J gamma gene, the VJ joining then happens, and this encodes the variable region of the T cell receptor gamma chain. So gamma chain locus. So how many gene segments are involved in generating the T cell receptor? So here we saw a similar picture or figure for B cells. So you can see here we have our immunoglobulin, our B cells, and our alpha beta T cell receptors. So here, remember, we talked about our kappa and lambda. So these are combining both options in our heavy chains. So we can see here that we only used kappa and beta and alpha, sorry. Then again, variable segments, D segments, our J segments. Again, here we can see our total diversity, with actually our T cells having more diversity. Or, sorry, with our B cells having more diversity. Yeah, you can see here 5 times 10 to the power 13 and 10 to the 18th. So the T cell receptor alpha beta gene rearrangement generates the T cell receptor. So rearrangements generate diversity in the T cell receptor alpha beta chains. We need, again, that VDJ recombinase activity with those RAG1 and RAG2 enzymes. The V to J gene recombination will have our variable region of the T cell receptor alpha. This will be transcribed and translated to form the T cell receptor alpha chain. So the beta chain, one D beta gene joins with one J. The variable beta genes join with the DJ genes. Then we get our VDJ gene recombination. And then we have our T cell receptor beta. This is, again, transcribed and translated to form the T cell receptor beta chain. The alpha beta chains pair after synthesis to form the T cell receptor. So this little graphic here is kind of just forming the process. So here's our beta chain forming down here, alpha up here. Now it's produced with our randomized V, J, and C. And then we have our alpha. Then we have a formation here, randomized again, and then our beta. Bad beta symbol. And then here's just this T cell. Or whatever cell it's on, but yeah, here's our T cell receptor. So again, as well in T cells, we have the 12-23 rule. So in immunoglobulin genes, D genes are flanked by 12BP RSS on both ends. So you can see red is 12. So here's our D flanked by 12 at each end. These D genes cannot be joined by D genes. So this is why it will bind to only a V or a J. So in T cell receptor genes, either beta or delta, D genes are flanked by 12 BP RSS and a 23 BP RSS. So here's a difference. We got a 12 and 21. So D genes can be joined by other D genes. So that's one of the differences. This generates additional diversity in the T cell beta and delta receptor chains. So the 12-23 rule is a gene segment flanked by a 12 BP spacer RSS can only join to a segment flanked by 23 BP spacer. Yeah. So you'll never see 12 to 12 or 23 to 23. These will not happen. It must follow the 12-23 rule. Similar to B cells, the diversity is concentrated in the areas needed most. So in the B cell, we saw that in the binding groove. That's where the greatest diversity occurred. So diversity arising from junctional diversity concentrates effects at the CDR3, the complementary determining region, as we can see here, stands for. And this is the location where a T cell receptor interacts with the presented peptide rather than with MHC. So T cell receptors interacting with peptide presented by class 1 MHC. So we have our CDR3, which you can see here. There's also alpha and beta of that. This is the hypervariate locations in the V region of the T cell receptors. And the T cell receptors concentrate their diversity in the third CDR. So we also, in T cell receptors, we also have combinatorial joining, where we have combinatorial rearrangement of the V, D, and J genes, which results in further diversity. We also have random sampling of the alpha and beta chains. So the alpha and beta chains pairing contributes to diversity. We can note that somatic hypermutation does not occur in T cells. That's important to remember. And again, here I can show this is a variability graph. We can see that CDR3 has the most diversity, and this is where the changes are focused. And here are the different types of diversity that occur. There's also insertional diversity, where nucleotides inserted at the V, D, and D to J junctions, aka the N nucleotide action, or the P nucleotide addition, We also have junctional diversity, where we have imprecise flexible joining of the V, J, or V, D, and J. Note we can have D to D and D to D, D joining in the beta and gamma chains. So this allows for the diversity within these two chains, the beta and delta, which again we saw in the T cell, in the beta and deltas, we can actually have 12 with a D and then a 12. So again, CDR3 with the most variability, and again, no somatic hypermutation in T cells. We also have the terminal deoxynucleotidyl transferase, or TDT, which carries out the N nucleotide addition in T cells just as in B cells. So a lot of the effector molecules like TDT, RAG1, RAG2 are all the same. So again, here's a figure of these factors contributing to diversity, as well as, our counts. So we have common order diversity and random alpha beta assortment, which results in 3 times the 10 to the 6 different alpha beta T cell receptors. We also have junctional diversity, results in 10 to the 18 different alpha beta receptor T cell receptors. So now we're going to be looking at the B cell receptors and antibody in more detail. So we've seen this figure before, and it's just a simple showing that antibody can bind to bacterium and can result in effector cells releasing, which are the plasma cells for B cells, releasing antibodies, which can bind to further bacteria. So each T and B cell is specific for one antigen. We've said this many times, but actually one epitope, which refers to the specific portion of the antigen these cells recognize. Here we can see that. So each T and B cell expresses a copy of its unique receptor on the cell surface. The adaptive immune response involves the activation of lymphocytes when they encounter antigen for that they are specific for. So what happens when a B cell encounters an antigen? This will result in multiple step activation process, and for T cells, this requires antigen presentation by APCs. These activated lymphocytes then proliferate, and only the lymphocytes specific for that antigen are activated. So here's kind of our whole repertoire. These are the ones that are specific, and these are the only ones that will activate. And these will release effector molecules. So we have a differentiation step. So in this differentiation step, we get our activated T and B cells, which differentiate into either our effector cells, which act immediately and cause antibody production for B cells and cytokine production for T cells. And then we also have our memory cells produced for our long-lived adaptive immunity. So here we have an animation. Move slowly. So here our cells are producing the antibodies. So we can see our receptors. Here's a virus coming in. So in general, this In this animation, we have a B lymphocyte with an appropriately fitting B cell receptor normally only reacts with epitopes of an antigen having a corresponding shape, which in turn and eventually enables the B lymphocyte to proliferate into a large clone of identical B lymphocytes. And these are by Gary Kaiser, which you can find his website, and there's a ton of these animations. So before T and B cells encounter antigen, they are referred to as naive B cells, or naive lymphocytes, They already have their specificity and their receptors, so they're already going to bind to one thing for that one antigenic epitope, but they're in a resting or quiescent state until they encounter the antigen they are specific for or presented to it. So B cell differentiation and clonal expansion is antigen driven with some T cell help, usually with T helper cells. So we have our memory cells, which react to the exact same antigen we encountered again. So the same one that causes initial differentiation into our effector cells as well. But during our memory or secondary response, where our memory cells are activated, we have a faster response time than our naive lymphocytes and usually more powerful as well. So we talked about this figure before with our primary secondary response. So our primary response is the first encounter with the antigen, where there's slower kinetics and the antibodies made in limited amounts. In our secondary response, we have our second encounter with the same antigen, and there'll be a faster and greater magnitude response. The response to the new antigen, so here let's just say we see another antigen while this is happening, we start the kinetics again and have another primary response. The amount of antibodies also in the secondary response is also higher. So again, it's not just faster, it's stronger and more effective. So antibody specificity. Antibodies made against a pathogen are highly specific for that pathogen. So antibodies specific for measles virus do not recognize an influenza virus. They just have their one target. So here's an example. Here's the measles virus. We have our antibodies designed to bind. Here we can see binding, a bunch of different spots. Same antibodies that are, let's say floating in our blood. All of a sudden we have influenza virus. Nothing happens. They will not work. And this works for pretty much anything that's not the measles virus. So antibody structure basics. So the antibodies are a tetrameric protein which have 4 chains, 2 heavy chains and 2 light chains. So here we can see our heavy chain, light chains. So then we'll have another heavy chain and a light chain here. So just write that. Sorry. Then we have our 2 light chains, like we said. Antibodies, or you'll see them referred to as this quite often, are produced and secreted by B cells, but some serve as the B cell receptor. So the actually secreted antibody and the membrane-bound version are pretty much identical. Antibody molecules entered in the B cell membrane are, so you can see our membrane-bound, there's a slight difference, but it's essentially the same by a short transmembrane domain. B cells and antibodies do recognize free antigen, but they do not need to see antigen presented by MHC proteins, unlike T cells. So as we discussed, T cells need to be presented through MHC in either CD8 or CD4 positive T cells, where B cells can just see it freely. But B cells do need T cell help to become fully activated by antigens and make antibodies. So this allows them to proliferate and class switch and other stuff, which we'll get to. The exceptions to this are sometimes that we have T-independent antigens, which can stimulate B cells without T cell help. So B cells recognize macromolecules on the external surfaces of pathogen. That's important, as we'll compare them to T cells at some point. So antibodies bind to shapes or epitopes on the surface of antigens. These antigens are usually proteins, which we said are highly antigenic or immunogenic. But antibodies can also recognize conformational shapes on other macromolecules, such as carbs, glycoproteins, and glycolipids. So here we have a picture of a bacteria binding to a B cell receptor, which induces a receptor-mediated endocytosis. This B cell engulfs and destroys the bacteria, or tries to, and the B cells can also process and present epitopes to T cells. So as we said, B cells are also APCs. So this B cell encounters a pathogen, takes it in. Now this B cell can present it on an MHC1, and now our T helper cell can come in and see it, and then now we can activate our T cell response and also induce better B cell activity. So antibodies recognize portions of antigens, the epitope, aka antigenic determinant. These surface epitopes can be linear or conformational. So linear epitopes are continuous epitopes. So a single segment of an antigen or polypeptide chain has a continuous amino acid sequence, so we can see that here. These fragments of antigen are peptides. Conformational epitopes which are discontinuous, are made up of amino acids from different parts of an antigen. So here we see our discontinuous epitope, which are made up of amino acids from different parts of an antigen, such as a polypeptide chain or glycoprotein, that have been brought together by protein folding. So here you can see the folding occurring. They are discontinuous in amino acid sequence. So here we have our linear epitope of a protein antigen is formed from continuous amino acids. A discontinuous epitope is formed from amino acids from different locations in the polypeptide, brought together when the protein chains fold. So a brief history for antibodies here. So antibodies have both variable and constant regions. The diversity in the variable regions posed a challenge for early research or immunochemistry until a source of a homogeneous antibodies was attained. So example, we use plasmacytomas, which is a plasma cell tumors for multiple myeloma patients. These produce large amounts of identical monoclonal antibody, which are all specific for the same antigen. So they had homogeneous structure. So essentially it's a tumor cell that just would not stop producing antibody. And this allowed us to determine that we have our light chain, heavy chain, and disulfide bonds. We also have a hypervariable region, which we'll discuss later. So antibody structure. So here, this is the protein structure, but I'll just draw it similar. So we have our Y. So here are the Fab. As we see here, here's our FC. So here, and then we have our constant regions. And then here's our variable regions up here. But here's this is a more appropriate look if you were. So the image shows the structure of the FC and Fab regions and the constant and variable regions of the light and heavy chains. So yeah, you guys can check this out if you want to look at more interactive antibody stuff. So the light chains have two domains, the variable and constant, which we talked about in our diversity. So each domain is approximately 150 amino acid residues. We also have a variable light chain, which is the N-terminal half, which is highly variable. These are conserved regions and hypervariable regions within the variable region. We have our constant light chain, or CL, which is the C-terminus half. These are highly conserved, and there's two isotypes or classes. As we discussed, there's the kappa light chains and the lambda light chains. So here we can see a diagram of immunoglobulin showing the variable and constant regions of the heavy and light chains and the location of the antigen binding sites. So here's the antigen binding sites. So let's see here. It's also shown intra-domain disulfide bonds in the hinge region and associated carbohydrates. So yeah, this one's a little more complicated, but yeah, here are carbohydrates in our hinge region. This allows for flexibility, as we'll discuss later. So the heavy chains. The heavy chains also have a V and C regions. The V heavy is the N-terminal part again, which again is highly variable, and again is approximately 150 amino acids long, and it also has a hypervariable regions within the VH region. The CH region is a C-terminal half again, and is highly conserved. IgG has three constant heavy regions, so there's IgG1, IgG2, and IgG3. Each is 150 amino acid residues. There's also 5 isotypes in total, which we'll talk about later. So here are our 5 isotypes of antibody or immunoglobulin. So you guys can see some slightly difference in structure, but one good thing to remember is G is gamma, M is mu, delta is D, A is alpha, so that's an easy one, and E is epsilon. The reason to remember that is just, again, it's just terminology, but you'll see them, people change it up on every textbook is different sometimes. So we have our CH, constant heavy constant regions, so that's what dictates the type. And there are 5 types of constant heavy chains and 5 antibody classes or isotypes, as we said, M, G, A, E, and D. And here's our immune response. We can see in a primary immunogenic stimulus, we usually have M, then G first. M is always first, and we usually obtain a steady state. Then our adaptive response will come in with IgG. Then we'll have our secondary immunogenic stimulus. And then we see here, we'll still get our IgM initial response, but we have a massive IgG response from our memory cells. And these are much more effective and usually more specific to the pathogen compared to IgM, which isn't as specific, or isn't as effective. So basic rules of antibodies. 1, they need to recognize and bind antigen, and this is the role of the variable region. 2, they interact with effector cells and molecules which remove or inactivate the antigen, and this is the role of the constant region. So here we can see an IgG molecule. So with our antigen binding sites, disulfide bonds, and in IgG we have a hinge region. So the Fab fragments, which are the Fab stands for fragment antigen binding, which are two identical fragments. Each Fab binds the antigen, so we technically have two binding sites for every antibody. We have the FC fragment, which stands for fragment crystallizable, which is paired with two CH2, CH3 domains of the heavy chain. This interacts with effector cells and molecules, and then we have a Fab squared fragment, which is a dimeric fragment of antigen binding regions. There's also pepsin, which is a fragment with antigen binding ability, but cannot interact with effector cells or molecule. Antibody fragments produced by protease digestion have been used to define antibody structure and roles of components. Papain cleaves antibody into 3 fragments, and pepsin cleaves antibody into 2 fragments. So pepsin and papain are used to study antibodies, allowed us to isolate the pieces of the antibody and study them individually, which allowed for a lot of progress in research. So the hinge regions, which we mentioned in the IgG molecule. So the hinge region provides flexibility to the antibody molecule. So we can see here the bunch of different epitopes for the pathogen, and allows antibodies to bind. So here we can see a typical Y structure. It's okay, but this one needs to spread more, and this one is narrow, so this still allows it flexibility. Each antibody molecule can bind to 2 separate but identical antigens. This facilitates interaction with effector molecules, including complement and the FC receptor. Some variation between the antibody classes. So IgG, A, and D have hinge regions, while M, E have no hinge regions. So that's important to remember. And that's interesting because a lot of M and E, we'll talk about later, is more involved in neutralization in some cases, in complement activation, and then E is in eosinophil activation and some other stuff. But yeah, just keep that in mind. So each antibody arm binds to one epitope of an antigen. So on the pathogen surfaces, there are many copies of the same epitope. Here, 2 immunoglobulins with identical antigenic specificity bind to the multivalent antigens with both Fab arms. So you see, usually one epitope will not just be a singular piece, they'll usually be multiple. Many soluble protein antigens have several different epitopes. Here we have 4 immunoglobulins with different specificities, which are all being bound by different epitopes on the same antigen. So here's our similar situation where this thing expresses tons of this epitope. But we can also have bacteria have many different epitopes, which is normally the case, kind of both, they usually both occur. So we can have multiple, so these all have different specificities for different epitopes. So they're all working together at the same time. There's also antibody diversity in an immune response to an antigen. So antibodies specific for the same antigen can differ in epitope specificity. So although they're attacking the same bacteria, they can look at different epitopes. So here's an example here, one without an epitope, antibody 2, antibody 3. So here's just a little graphic of an antibody, which is pretty interesting. So again, we can see our FC, then our Fabs up here. Yeah, this is a good figure. So again, check out the website, same website we saw earlier, if you want more information. So immunoglobulin, or IG, aka immunoglobulin folds, the IG immunoglobulin domains, are repeating structural units found in varied cellular structures. So antibodies, T-cell antigen receptors, and some cytokine receptors and cell adhesion molecules, as ICAM and CD54, or CD54. Each immunoglobulin domain is made up of 7 antiparallel beta strands arranged in 2 sheets of 4 and 3. The 2 sheets are conveniently linked by a disulfide bond. So this is kind of structure showing the different beta-plated sheets. Again, another website to check out. So each light and heavy chain is made up of domains. So each domain folds into a similar tertiary structure, the immunoglobulin fold. So a stable structure found in many different receptors is the immunoglobulin superfamily. The immunoglobulin fold is a stable structure in the immunoglobulin superfamily, And these domains are held together by disulfide bonds, similar to the T-cell receptor. The light chains are made up of 2 immunoglobulin domains, or VL and CL. So each domain is a barrel-shaped structure with beta sheets held together by a disulfide bond. In the immunoglobulin fold, hydrophobic sequences are oriented to the interior, and the hydrophilic sequences are oriented outward, similar to our cell membranes. This promotes stability of the secondary and tertiary structure of the immunoglobulin domain. So heavy chain. The heavy chains are also made up of domains. There's one domain for variable heavy, and 3 to 4 domains for the constant heavy region. The constant heavy region structure varies with antibody class, as we said. This includes G, A, D, and D has 3 constant heavy regions. IgM, E have 4 constant heavy regions. So here we can see example, constant heavy regions 1, 2, and 3, 2, 3, but again, these have 3, these have 4. So our hypervariable regions. So complementary determining region allow for our hypervariable regions. So the complementary region have a major role in antigen binding. They help determine specificity of antibody for antigen. We also have framework regions, which are less variable portions of the V region. So here we can see our heavy chain V regions and our light chain V regions, and our variability plots. So this is a variability plot comparing amino acid sequences of the V domains in several heavy and light chains. We can see 3 hypervariable regions, HV2, HV1, and HV3, which we can see here. We can see the huge spikes in variability. And these are flanked by the less variable framework regions, as we discuss here in the blue. And then I also have hypervariable regions, or aka complementary determining regions, which are 1, 2, and 3, which we see they line up 1, 2, 3. So these hypervariable regions are located in discrete loops of the final folded antibody molecule. The hypervariable regions, or CDRs, are positioned on a structure of the variable domain, which form loops that converge into folded antibody molecule. So here we can see the hypervariable region in the variable domain. So here we can see the 3 different loops in our CDRs. And again, these match up to our 1, 2, and 3. So the hypervariable regions are located in these discrete loops in the final folded antibody molecule, as we show here. The hypervariable regions, or CDRs, are positioned on the structure of a V domain, which form loops that converge in folded antibody molecule. The heavy and light chain pairing brings the hypervariable loops from each together. This forms one antigen binding site in each arm of the antibody. So these basically are this little red area. So antibody molecules play a dual role. They recognize and attach to specific antigens, where this is the role of the variable region. They also neutralize or destroy antigens through biologic interactions, through the role of the constant regions. So our constant regions, also called the FC region, this is where our other cells come and bind to antibodies. So we have an antibody stuck to bacterium here, attaches complement, then the complement forks. So this is our anti-classical pathway. Then we have the formation of MAC, membrane attack complex. And if you want to review on that, check out lecture 3. Here's a macrophage through antibody-dependent cytotoxicity, binding to the FC region, and then this will go phagocytose the bacteria. Same with the neutrophil, binding to a complement receptor. So yeah, they're not just there to tag and work with T cells. They also work with a ton of innate immune cells. So the antibody structure is ideally suited, for these dual tasks. It enables precise specificity for antigens and conserves structures of biological activity. So guys, I hope you enjoyed that one. It's pretty interesting because antibodies are so relevant. And again, pretty much every vaccine and COVID, everything relates back to antibody immunity. Although T cells are vitally important, B cells are kind of the primary focus, as we know a lot more about B cells than T cells, although we know a lot about T cells. But if you look in a lot of textbooks, they pretty much got B cells down. So it's pretty interesting that we have such advanced knowledge of this topic. And B cells are antibodies are so important in so many medications and treatments that it's crazy what we've done. And there's still more to come, which is super interesting. I hope you guys learned something. See you next time.